I hereby open this academic ceremony in which Nicolò Mangraviti will defend the academic thesis LncRNA Bigheart in Cardiac Disease and Development. Dear candidate, may I invite you to present a summary of your study and the conclusions of your thesis?
Thank you, Prorector. Member of the Corona, family, friends, attendee online, thank you for joining me today. Today I will give you a brief introduction of what was my research throughout these 4 years, and the name of this thesis is Nicolò Mangraviti Bigheart in Cardiac Disease and Development. I will start with a very simple statement about the heart. The heart is the best athlete of our body. If you consider it a beat throughout all your life, constantly, without having stop, we can consider him as a very well fit and trained athlete. However, if you think about how the heart will actually carry this function, you can think about heavy lifting. It will do constantly throughout his life without you even notice it. However, less intuitively, you can say that all this skill coming from a genetic program. So a series of genes will be activated and will allow your heart to do exactly what it's supposed to do throughout all your life. You will never notice, but your heart will actually follow a specific program. However, even the best athlete can get injured. So upon injury, stress, fatigue, combined with comorbidities, the heart could actually not be able to carry the effort anymore. And as a good athlete will try to do, he will try to adapt to the effort. And intuitively, you can think about they start to try to increase his size for being stronger. However, in a lot of cases, this is not going to work, and the overcome will not function as we think. And that will be a cardiac hypertrophy, which is maladaptive. So the heart will grow, but the strength of the heart on the end of the day will be lower, and therefore it will not be able to carry the same strength it will have normally. Interesting to consider is that also in this case, the genetic program that will regulate this new stage will be different from its previous one. Considering that then we need to study this in order to understand how to cure such a condition, we need to first understand why the program changed and how can study this. So first we need to consider how the heart analyze these stimulations and how reply to them. You can think this in the simple way as an electric plant. We're going to have a signal which then is going to be broadcasted throughout several intermediators. And then we'll end up finally in changing the transcriptional level on the DNA level. So we have a signal which goes through different intermediators, and at the end it will change the transcription of a specific gene. So if you want to study all this modification, you can study either the signals, so you study the proteins and molecules that control these signalings, the intermediator, so all the proteins that control this passage between different pathways, and then the gene on the DNA level, so you can check which gene they altered when you have activation of a signal. But we're missing an important aspect of biology, which are the RNAs. I think that most of you, sooner or later in your life, you went through this dogma. We know that DNA goes to RNA and then goes to protein. However, if we look a bit more carefully, only a specific class of RNAs, which are called messenger RNA, are translated in proteins. But if we look on the overall amount of RNA our body produces, only a very tiny fraction is messenger RNA. All the others are considered non-coding RNAs because they don't produce protein. And these are like the 4 main classes we know. They are not all, and not necessarily the most abundant in our cell, but the one that we actually know slightly more. Now, for making an example to how important those classes are, let's make an example with one of the most commonly studied, which is macroRNAs. Let's think about our plant, and let's think about what will happen if a macroRNA works. So what the macroRNA will do, it will actually reduce the strength of a signal, and the signal that you're going to have at the end of the day will be lower than the previous one. Then, of course, can actually be higher or lower depending to your target, but the logic will be always the same. One single function which can actually dramatically change how a signal will be broadcast, and which will be the end of this. However, as I say, my thesis involved non-long coding RNA. And the reason why I'm so fascinated from non-long coding is because you can consider that as a Swiss knife, has several functions and is a multi-tool. But how can we study them and how that influence the heart? let's take again the example I gave you earlier, and let's take in consideration all the possible functions that can have. In one situation, we have a signal that change accordingly with the signal the null encoding will give. For instance, in one case, the null encoding can boost a signal. In one case, it can actually weaken a signal, or it can skip completely these two aspects and altering the transcriptional level of your DNA. So it will change the expression of some gene. So as you can see, it's already 3 functions against 1. So it's already different. But how does it do this? That's an idea of how a null encoding takes shape in the cell. It's a 3-dimensional structure. The 3-dimensional structure of a null encoding allows it to interact with protein, RNA, and DNA, creating a very sophisticated complex, which will then determine which is the function the null encoding will have. Now, not necessarily you're going to have all this together, but you can have combination with different protein, different RNA, and different elements of the DNA level. So the number of combinations will be pretty much unlimited if you consider all the different variations that your protein and your RNA can have. But how that actually get in the picture that we actually try to design today? Think about your heart. Now, research showed that some null encoding can actually be harmful for your heart upon cardiac hypertrophy, actually inducing it, and some other can actually shield your heart from such an attack. So you can see a null encoding as a sort of like balancer. Some can be actually beneficial, some can be not, depending to the situation which we study this. So if you consider what I tell you earlier, and we know that every of these will have a different program, we can also consider that also we're having a different set of null encoding RNA which regulate cardiac hypertrophy. So the main goal of my thesis was originally identify novel null encoding RNA in cardiac hypertrophy. For doing this, we applied 2 different mouse models. One, we induce cardiac hypertrophy through surgery. We're using transverse aortic constriction. And the second one, instead, it was a transgenic mouse, which we boost the expression of a specific gene, which will end up activate the hypertrophic pathway. That's what, in fact, we saw in our histological analysis. And then we checked the differential expressed gene between null encoding RNA, in these particular cases. But we need to consider another important aspect. Although they are both cardiac hypertrophy, they're not going to be identical because they are 2 different models with 2 different systems. But in this case, we look for similarities. So if you consider that, for instance, in a grocery shop, you will having a similar brand and similar price, but they're not going to be identical, that's what you can expect. We're going to looking for things that are similar between the model, not for the differences. And looking for all the things that are similar, the top candidates of our analysis was this null encoding called Bigheart. So considering that we saw both overexpressed in both models, we suspect, okay, he has to be involved in signaling in some extension in cardiac hypertrophy, but we don't know how. So, starting with this idea, we start to considering which are the main pathways that regulate cardiac hypertrophy. And one of the strongest pathways and the most well-known is the NFAT calcineurin pathway. So I simplified this as much as possible, and this pathway has 3 main intermediators: calcineurin, ERCAN1, and NFAT. And the final method will activate hypertrophic-related genes. However, we don't know if Bigheart interacts with this pathway or if it regulates a different one. So we overexpress Bigheart in cardiac cell using adenovirus association virus. So we overexpress with a virus Bigheart in cell and see what happens. What we notice is that upon overexpression of Bigheart, we have a significant increase on the translational and post-translational level of ERCAN1. So one of the intermediates of this pathway raised in expression significantly. So we can consider this as a change in what we saw earlier, and if you think about what I showed you earlier, this can be a booster in the signaling, increasing the transcription of those genes which are related with cardiac hypertrophy. The next question was like, okay, we know that Bigheart is involved, but who is going to regulate Bigheart then? We figured out that NFAT, another intermediate of this pathway, regulate Bigheart itself. So we start to analyze this as an internal loop in this specific pathway. And the final model we come up is something like this. The signal being boosted on ERCAN level. However, once arrived to the DNA level, activate Bigheart, and then the circle starts again, boosting the signal over and over, and self-maintaining cardiac hypertrophy. And that was actually a general idea of my chapter 3. However, it's not that simple. So far I give you an idea that we can study signaling studying as an electric plant. This is not completely true, because if you think about all the signaling that your cell will actually control and govern simultaneously, there are several possibilities, several interconnections between several pathways. So the reality will be a bit more complicated. So how can I study this? In order to study this, we apply chromatin immunoprecipitation on RNA, which is a technique which allows to study a bit more in depth what a null encoding will do. Simply put, it's like a magnet. We're going to have a magnet which is able to attract specifically your null encoding RNA, and by pulling him, he will pull everything is actually associated with this null encoding. Once we remove the magnets, we can separate the RNAs, the protein, and the DNA associated with our null encoding. Now, using this, we can identify the network which this null encoding is involved with. And the network we identified was actually around 500 genes. Now, not all these genes will work together. Like 500 genes working for the same pathway will be possible, but it's not what we are looking for. So we're using a system of clusterization. Some genes will be involved in the same pathway, and by using bioinformatic tools, we actually identify which are the commonly genes associated with this. And the most overexpressed pathway were the mTOR signaling and the GnRH. Both are involved in cardiac hypertrophy and in cell development, which was very interesting because the two are not completely separate. Another aspect we can also check is like, okay, we know what happens if we have Bigheart. What does it happen if we don't have it? So we're using a system which actually allows you to destroy the null encoding using GapMir in cells which they go under cardiac hypertrophy. What we notice is that the genes that actually are differentially expressed are all involved with development or morphogenesis. And this was very interesting because you need to consider that the heart needs to actually develop throughout its life, and once it's fully differentiated, it's not going to do anymore. So, but for give you an idea what this means, is that every time you have a specific stage of your heart throughout its develop, you will having a specific genetic program, similarly to the model I showed you earlier. Every moment and every different step will have its own. And once it's fully developed, some gene will not be necessary, so will be shut down. And interestingly put, we found that Bigheart does exactly the same. It's highly expressed throughout the development of the heart, and once the heart is fully developed, it doesn't be expressed anymore. And we found this both in human and mouse, so it was conserved with the species and was pretty interesting. But so far, we just given bioinformatic information. We need to prove this in a model that is a bit more on bench side. So we using a system for cardiac differentiation in vitro using iPSL. So iPSL are cells which are not differentiated yet and which you can differentiate in specific cell type. In our case, cardiac cell or cardiomyocyte. We differentiate this cell and took for every step the halfway. So we took half differentiated, full differentiated, or later level differentiation. What we notice is that the level of Bigheart drop throughout this in vivo differentiation. Adding another level of complexity to this picture, we also know that Bigheart is involved in several mTOR genes, we showed you earlier. So we check what happened to the signaling. And we noticed that the signaling of the mTOR pathway reduced in strength throughout the differentiation, following or completely inverted what we saw for Bigheart. So we came up with a different model in which when Bigheart is active, it boosts this signaling, activate the mTOR pathway, and we're having our cardiac differentiation and cardiac development heart. However, once the differentiation is gone, Bigheart shut down itself, and the strength of this signal reduces itself in a fully developed heart. And that's already been shown also in literature, so it was perfectly what we were expecting to see. So, and that was actually the general idea of my chapter 4. So, in conclusion, throughout this presentation, I give you a general idea of how the heart is regulated, how a null-encoding function can actually regulate some aspect in heart physiology, and how a novel null-encoding RNA can be important in regulating cardiac hypertrophy through a specific important pathway, and how the same null-encoding in a completely different scenario is actually beneficial for your cell because it helps them to develop the heart. And if you think about it, the same null encoding can be, in some case, good and bad. This reminds me a bit of like the concept of yin and yang, in which interconnected forces can be beneficial depending to the aspect and the moment we're looking at this. With this, I finish my presentation, and I give you the word back to the prorector.
Thank you very much, dear candidate, for your very clear and educational lecture. After years now, I finally understand what your promoter does. So this is really good how you presented this. The opposition now will be opened by Professor Baker. He's Professor of Cardiovascular Sciences at this university, and in addition, Chair of your Thesis Assessment Committee. Professor Baker, please.
Thank you, Prorector. Dear candidate, congratulations on a very nice presentation and also a very well-written, thorough thesis, and also congratulations to your supervisors providing you with the environment, supervision and mentorship to produce such a nice thesis. I wanted to start by just asking one of your paranymphs to state Proposition 2, please.
Long-non-coding RNAs can have multiple roles depending on the context. Quote this thesis.
Thank you. I think you've illustrated that a little bit but can you expand a little bit on your thoughts here? You have a long non-coding RNA that seems to be in the heart studies and also in the IPS model to be expressed in development. Yet you have something that's also involved in hypertrophy and disease. Can you sort of marry those up a little bit more about the molecular mechanisms that you think are responsible for that? For example, different binding partners, different activation. A little bit more detail would be useful.
Hi, esteemed opponent. Thank you for your word. And for answering your question, so as you were mentioning, this null encoding has different functions between hypertrophy and differentiation or development. And mechanistically speaking, we identify several proteins through mass spec that interact with this null encoding. One in particular that we found pretty interesting was HGB1, which is actually pretty much a protein that actually helps to bend the DNA. And if you think about mechanistically speaking, that could make much sense for a null encoding that works in trans. Now, we talk about cis and trans, but what actually the latest paper about say that cis and trans doesn't exist. It's speaking more about three-dimensional structure of the DNA. So a null encoding can actually bend the DNA in some specific point, can be very beneficial because it will actually make enclosure some area and allowing the condensation of polymerase in that area, increasing the transcriptional level of those genes in a more proper fashion. We also know that now we call it the super factory or polymerase factory, which we have the aggregation of several polymerases in the same area, and they work like in parallel for transcribing different genes, which usually are commonly associated with the same pathway. Actually, that's actually could fit what we saw. So we think that in this particular case, HGB1 is one of the main effectors of this, although we did identify several HNGBR proteins. But that's a bit more tricky because when you're using technique as a chirp, you always have an over-characterization of those proteins because, of course, on DNA you have all these proteins set around. That's why we focus our attention on identify, which we could also identify by Western blot. Because in that case, the level of the enrichment of that protein will be so high that it could be significant for the function of the null encoding itself. Furthermore, HGB protein seems associated with the triplet of G elements, so triplet of G, like a big number of G in the null encoding area. And Bigheart, especially in its 5 termini, show a considerable amount of this G, suggesting a possibility also conserved area which the protein will associate with, given also a context in the functionality of this null encoding and the elements.
Okay. I noticed on your in your IPS model where you differentiate to cardiomyocytes that the expression level of Bigheart is highest in the pluripotent cells, right? And it goes down. Mm-hmm. So have you thought about the role in pluripotency?
Yes and no. I do see the same, but I have to admit that if you compare the level of mouse differentiation and human differentiation with the one we find in iPSL, there is a little discrepancy. With the iPSL, you see this nice drop throughout the differentiation per se. But if you look on RNA data from different databases, the level of Bigheart starts to drop after day 1, so when the heart gets fully differentiated and does not replicate anymore. So I do suspect, and that's actually only my thought, that although it can have an effect on the activate similar pathway during the differentiation, its main function is maintain activate the program of differentiation and then shut itself down. However, in a model like in vitro, this will not be perfect to analyze because it's actually still a bit artificial in some extension. So as much as you can reproduce the differentiation, will not perfectly mimic the real situation.
Okay. My next question was really around conservation of LncRNAs. So you've worked from the mouse model, essentially. But there are quite a few studies out there, particularly from people like Norbert Hübner, systems level, to show that the main conservation in LncRNAs is between primates and humans, and then less as you go to different species. But you've started the other way around. I mean, is it your belief that long non-coding RNAs that are conserved across species are more important than the ones that are just between human and primate?
Well, I think it depends a lot from, I think in this case we need to talk about a level of complexity of the organism. Now, we know for sure from, I don't remember the year of the paper came out, but roughly from the earlier stage of differentiation, we have 756 non-long coding. They are perfectly conserved, like almost outside the vertebrates realm. However, if we look from the one they are completely associated only with human and mouse, we're around 5,000, if I'm correct. So I think that those non-long coding, they are involved in some common pathway which is actually specific for that specific species. So we don't need to alterate their pathways necessarily like that, so we maintain the structure like that, a bit more like protein. In this case, we think about more structure than sequence, but the logic of maintaining the non-long coding in an area which can regulate the DNA will be the same. We just given a less thought about the sequence and more about the structure.
Okay. I had a technical question. Your siRNA studies, you use a single siRNA.
Mm-hmm.
The field normally suggests that you need to replicate that with a second siRNA for reasons that you probably well know. Did you do that or not?
The RNA sequencing with gap mirror, I guess, right?
No, the siRNA studies where you interfere with Bigheart and you knock it down. And the gapmer studies you've done with a single sequence that you've designed and tested. But normally in the field you would replicate this with a second siRNA because siRNAs have quite a common off-target effects.
Sorry. I think that the most logical way to repeat the experiments will be use ASO. And another technique we could apply will be CRISP inhibition, which will give you an even more clear way of how this non-long coding will work on the transcriptional level. Because if we are correct and this non-long coding bend the DNA, that means that it can start to assay with the protein as soon as it starts to be transcribed from its locus. Although we're working for a trans non-long coding, however, reducing this transcriptional level without altering the locus, we could actually see what happens if we don't have this transcription whatsoever. So I think if I have to repeat it, a better way would be actually CRISP inhibition for having an idea if it is co-transcriptional to non-long coding. However, I will also add another level of complexity using also ASO for see what happens if it's only the full length of the non-long coding that is action, or also co-transcriptional.
Okay, thank you very much, and I'll hand the word back to the prorator.
Thank you, Professor Baker. The opposition will now be continued by Professor Fatica, and he's Professor of Genetics and Molecular Biology at the Sapienza University of Rome. And he's also a member of your thesis assessment committee. And our university expresses its gratitude to you for being present here at this opposition today. Professor Fatica.
Well, thank you. Nicolò, nice to see you again after all this year. And I have a couple of questions about the mode of action of Bigheart. The first question is, how did you exclude the Bigheart act in cis, actually?
Hi, Mr. Point. Thank you for your question. When we actually checked by CHIRP, we didn't find an enrichment of peaks around the two neighboring genes, which are EGFR1 and SIMS. And we didn't see a significant differential expression between the two of them upon Bigheart overexpression. So either the level of overexpression is not significant enough for see it, or is not involved with that. However, I think that a better way to see if he's still working in CHIS will actually do a single molecular RNA fish and see if it's only in his locus or it's actually propagate around. But we didn't go for that way yet.
But if you overexpress a non-coding RNA, you cannot discriminate between cis and transacting function. Actually, because if it's overexpressed, it's not going to be transcribed on the same locus. So that's why I asked my question. So the overexpression is not sufficient to say that the London coding is not acting in cis. You know what I mean? Because you are expressing the London coding from a virus. It's episomal, it's not integrated in the genome, it's not in the same position as the endogenous London coding RNA. From your thesis, there are no data that exclude the cis-acting action. For example, when you treat the cardomycete with the GAPMers, did you analyze the IGF-1R expression? Because by CHIRP, we are looking to the stable non-coding RNA molecules. But it is possible that it acts only by transcription on IGF-1R, for example, which is very close and antisense.
Yeah. So a better strategy, I think, will actually fit the same question Professor Baker was making later. So I think that for checking if it can work only in cheese without altering in any way the locus, CRISPR activation or CRISPR inhibition can actually give you a better, a more comprehensive picture of how that works. However, I was also considering the level of diffusion of the non-long coding. It's true that you were expressed the non-long coding with an ectopic vector, whatever it's going to be, retrovirus, overexpression with virus, it will be from a different area. However, it's been noticed, for instance, for PVT1, that PVT1 stays in the same locus and is known to be working only in cheese, so it doesn't diffuse throughout the cell. So I was suggesting single molecular RNA fish, because in that case, if it's working outside this locus, we're still going to see a sort of diffusion around. But a more appropriate way will probably be CRISPR activation or inhibition, which will give you a more precise picture of what happened on its transcriptional level.
Or the insertion of a pass. If you
True. Also the twister system.
Yeah. The second question concerns the subcellular localization. Did you check by fish, as you were saying, the subcellular localization of the endogenous big heart and of the overexpressed big heart?
No, we didn't do the variation between the two. We just overexpress Bigheart and see what happened in total without differentiate between the overexpressed isoform and the inside the locus of it. It could have a differential structure, that's correct, because of course we have post-transcriptional modification which happen co-transcriptionally, so we might not mimic then in a different model. However, the fact that was interesting to us, it was still able, even in this way, to overexpress a specific gene. For us, that was actually interesting, because if it was do, for instance, N6A post-transcriptional modification or interaction with some element like proteasome, we're not going to see that. But also, topically speaking, we have a very strong effect on both transcriptional and co-translational level, which for us was actually meant that he has to be the full length to do most of these functions.
Okay, for me it's okay. Thank you, and congratulations, Nicolò.
Thank you, Professor Fatica. The opposition will now be continued by Professor Condorelli. He is Professor of Cardiology at the Humanitas University of Milan and also a member of your assessment committee. And also our university expresses its appreciation to you, Professor Condorelli, for joining us here at this opposition today. Professor Condorelli.
Thank you. Thank you for the presentation, which was very complete, and the work that has been really, quite complex, taking care of a lot of aspects, etc. So, congratulations. I will have a question. So, regarding the human counterpart of your non-coding RNA. So, if you could tell us more about the human, whether you have identified the human homologous of your non-coding, and what is the degree of similarity with the mouse, because this is not clear. And secondly, how you envision the application of your non-coding RNA in therapeutics. Also because I saw from your data that you have identified this non-coding RNA also in liver and other tissues. So I was just curious to know how you would basically think that this non-coding RNA would be applied for therapeutics through any specific method or just cardiac release or I don't know, to other methods that you may tell us about. Thank you.
Hi, esteemed opponent. Thank you for your word. So, I have to give you a bit of background on that. Yes, we did study mostly the homologue in human and mouse, which was our main target. And when we considering this, we need to actually starting with the point that the size of the null encoding is different. We have a null encoding, which actually now it's been described as a 3 isoform system, which has roughly 1,000 base, 1,000 base pair. Instead, the human null encoding has roughly 573 base pair. So it's already a difference in size. However, what we used in order to validate its presence and its identity was a model of prediction of two-dimensional structure. We know that for null encoding higher than 1,000 is a bit less precise, but for null encoding between 500 and 600 is pretty precise. And we noticed that there are striking similarity between the two of them. Additional level of complexity will actually given from the position. So they are in the same value locus with similar distance between the two genes, both in human and mouse. And they show a similar trend of expression throughout differentiation and throughout the mechanism. So there was another additional level of complexity and another validation that that was the right isoform that we were looking for and the right null encoding. That's how we select that as the most precise, or at least the most valuable candidate for the Bigheart counterpart in human. For a therapeutic approach, I will think that we cannot use directly Bigheart. What we can use, though, is structure and functional elements. Because if you consider that we could, and that's actually still speculative because there is not a system like this yet, cut null encoding Bigheart in several parts, see which of them interact seems with the same gene and with the same protein, and then we could engineer those elements in a system a bit more controllable. Because you were mentioning null encoding Bigheart RNA in mouse is expressed in several tissue. And this would already be a problem for a specific targeting, heart-specific therapy. What we can do that will be using a specific system of delivery which is localized only in the heart without altering the pattern of transcription in other tissue, because we don't know what this null encoding does in those tissue. We know that it's highly expressed in heart throughout the differentiation and development, but we don't know what it does in those tissue. We could do the same since it is involved in similar pathway, but we cannot verify that because we didn't do experiments in that. So the system for studies will actually identify functional elements, isolate them, and then using a specific system of delivery for the heart in order to control or manipulate a bit better its function. In our case, silencing will be the best technique because, of course, we see that this is actually highly expressed in cardiac hypertrophy. So if you shut down Bigheart, you should have a reduction, as we show in some of the experiments. However, we don't know where to target, because if you have several isoforms and we design the probe on the wrong isoform, we're not going to block at that. So study the functional of Bigheart will be the first step. Design a specific probe or specific ASO for that isoform will be the second step. And finally, canalize all the information in a specific system of delivery, which is target specifically the heart in a condition of stress.
Thank you. Thank you.
That was it, Professor Connolly?
Yes, I would say so, because I saw that you have already started working on IPS cells, human cells. And I also seen an effect the mTOR pathway, mTOR pathway. So I guess that you have asked yourself how this non-coding RNA would work in the human context. Unfortunately, the iPS cells are not really ideal because they are fetal cells. So it's really tricky in the sense that we cannot just say that this is a differentiated human cardiac system. So maybe in the future, the human hearts slices or systems like this would be ideal for testing molecules that were identified from mouse or other approaches before going to clinical trials. But this is a common problem to everybody who is working on cardiac experimental cardiology, basically. So, but I see that you have worked out these problems, and quite well what are the problems related to this type of research. So I was just curious, in the future, what do you think, where you see yourself? So in continuing working on this topic, or having experienced this type of research, where do you think that you will work in the future? In the same topic, other topics, cardiovascular, cancer?
So at the moment, I'm doing a postdoc in the University of Yale, working on non-coding RNA as well. However, in the future, I will try to something a bit more translational, because I always been fascinated between the striking similarity of transcriptional effects, especially in the early stage of differentiation, in brain tissue and heart tissue. So I wish to working on how non coding can actually regulate the switch. That will be my future direction. And the functional elements and mechanistic effect of a non coding has always been my main target. So I think I will keep working on that, but in a more translational way.
Thank you.
Well, thank you, Professor Gondorelli. The opposition will now be continued by Dr. Gladka. She is Assistant Professor of Molecular Cardiology at the Amsterdam University Medical Center. And also to you, the university is thankful for joining us here today in this defense ceremony. Dr. Gladka.
Thank you very much. First of all, dear candidate, I would like to congratulate you on the beautiful thesis. I enjoyed very much reading that. And I have to say, the study in cardiac hypertrophy, it's really close to my heart since I did my PhD in exactly the same lab. So I'm really happy to be here. So I would like to start with therapeutic applications, as Professor Condorelli already started, and you explained very well how would you do that. But the patients that have a cardiac problems, they already have developed hypertrophy often. So I was wondering if your Bigheart would be able to revert that phenotype. So you did a study where you actually delivered or inhibited a Bigheart in a heart and then induced the hypertrophy. So do you think this would also work as a therapy? So in already diseased heart, if you would treat, would it help or not?
Hi, Talent. Thank you for your word. So a system that actually delivers system for silencing the non coding itself might be beneficial for a patient, because we also know that Bigheart is highly expressed, and I showed earlier, it boosts a significant pathway in which we know it's one of the main factors in cardiac hypertrophy. Although, in order to do that, you need to consider a different aspect. As I was saying earlier, we know now that at least in mouse we have 3 isoforms. In human, we still see 1. So the first things will be actually logical to do, it's identify if there are additional isoforms in human. Once that is identified, I would say that ASO will be the way to go. So I don't know if the straight application will be the logical way at this stage. Probably using organoids for verify in a condition of stress of the organoids will actually be a more appropriate way to see what happened on the physiological level. But I'm not so sure about this. It's only quite speculative, this. So I would say that the best idea will actually design a series of bigger targeting probes, or ASOs, and see what will be the effect in a condition of hypertrophy. That would be the way to go for application in human, because in that case, that's the main point, like Bigheart is overexpressed during these kind of conditions. However, and that's the part that is a bit more tricky in my mind, is that like, when you actually have a cardiac hypertrophy, you need to also consider that you have a sort of like a regression of the heart. So the heart goes slightly back because can reactivate a lot of genes which they were deactivate, and fetal gene they are reactivate too. So also Bigheart does that. And the point is that, does Bigheart reactivate as a maleficial effect and that's it? Or is because the heart will reactivate the program because it starts to bring itself to a fully differentiated heart? So silence Bigheart in a patient might be completely different from what we saw in vitro. That's why I was thinking, let's first analyze how it works mechanistically speaking, and how can we use that mechanism to our advantage.
Okay, right. So you did not consider performing study in mice where you first induce the TAC, let's say, and let the heart develop hypertrophy and then do your treatment?
Yeah, there will be actually like a very great idea. However, and that's again the problem, the level of Bigheart in different tissue in mouse and human, that could be a bit of a problem because it might have additional function that doesn't have in human. So doing all the testing only in mouse could give you a fake positive or a fake negative, because we know that in human it's highly expressed only in brain and in heart, and even with that, the level of transcription is very low in fully differentiated tissue. So we should see what happens throughout differentiation, and then we can forward thinking how to do it.
Right. Okay. And I was wondering about the mechanism of activation of a Bigheart. So you nicely show in your thesis that the calcineurin end fat is involved in activating a Bigheart in an adult heart, but also in your figure 1 from chapter 3 at page 75, it was already mentioned that in your IPS experiment, you see around 200 fold increase or increase of this long non-coding RNA compared to the late differentiated cardiomyocytes. So my question is how the Bigheart is regulated in this developmental stage. So I know, or I just actually read today that the calcineurin end fat is not necessary for development at this stage. So do you have any idea what is regulating this long non-coding RNA during early differentiation?
So the idea that I came across was a bit similar to my first answer to Professor Andrew Baker. That's why it was so fresh in my mind, because I was wondering tonight, to be fair. So I do believe that non-coding RNA, especially the one associated in differentiation, they're going to alter the structure of the chromatin around the locus they're going to regulate. I also think that this particular non-coding band DNA allowing a more precise three-dimensional structure of the genome in throughout the differentiation. Now, why this could be useful to the cell? We know that throughout the differentiation, the program changes. So we also know that the differentiation will be activated and then deactivated over time. If you think about this something that regulates two-dimensional structure, higher level of transcription of this non coding could actually have a tinier or like a different structure of the DNA. And the lower the level of Bigheart will go, the less will be able to affect this three-dimensional structure, altering the transcription of all these genes throughout the differentiation. So I think that if we think about something mechanistic, that's the logic. Altering in a very selective way the three-dimensional structure of the DNA, allowing or blocking, in some case, of course, transcription of specific gene by altering over time the overall three-dimensional structure of the genome. And that will make sense with the drop, because I don't need those gene transcribe anymore. So probably I need then to actually get loose for getting outside the factory, the polymerase factory. So the less Bigheart will actually bend less the DNA, so can actually shorter or increase the distance between those areas. That was actually my first guess. But in order to verify that, I should use something like 4C in different level of differentiation and see if that will actually work like that. But we didn't follow the experiments yet.
Okay, thank you very much. I think I do not have more time, so I will give word back to the prorector. Thank you very much.
Thank you much, Dr. Gladka. The opposition will now be continued by Dr. Calore. She is assistant professor of molecular genetics at this university. Dr. Calore.
Yeah, thank you. So, dear candidate, I really enjoyed reading your thesis, so I would like to congratulate you and your promotion team for the support over these years. So, I have more technical questions for you. In the first part of chapter 3, you describe the structure of Bigheart, and you first use the minimum free energy and the RNA fold, and then you switch to shape sequencing because you say that there is the first approach that you use is suboptimal after a certain threshold of length. So I'm not familiar with this topic, and today I would like to learn something new. So I would like to know what is this threshold after which the sequence is too long for the prediction being reliable. And also I would like to know more about shape sequencing. So what are the main steps? What are, for example, the quality controls that you use, if they are the same as RNA sequencing, and yeah, some more technical details about these two approaches that you used.
Hi, esteemed opponent. Thank you for your question. So, depend a lot from which is the program you use. At the moment, if I remember correctly, the state of the art said that between 500 and 600 nucleotide, you could have a relatively appropriate three-dimensional structure. The main problem with this resides in the number of variations you can have. If you think about the three-dimensional structure is depending from how many shapes a single null encoding can take. But the longer it is, the more likely is that the number of variation of that shape will change. So the larger number of possibilities will happen. Not all of them will actually be allowed on the three-dimensional spectrum because the entropy will be too high in some extension. But nevertheless, the number will raise significantly. So at the moment, the algorithm can actually identify as a more likely, not 100% sure, but the more likely until 500, 600 base. That is the threshold. Although, like recent paper, yes, I think now it's actually in printing, they say that they could boost that until 8,000, 9,000, but they couldn't take in account the modification, which again alterate the three-dimensional structure and then for the number of forms. But at least that is work. For about the SHIP-seq, I did not did myself. However, as a general system, we know that the cell being treated with, I don't remember the name of the molecule in this moment, I beg your pardon. However, the number of reads will be changing accordingly for the closer to each other. And then you can actually identify the structure based on how the distance and how many signals you can get from the same nucleotide in the same locus. And that's why you're having different color also, because the number of reads for that specific, in that specific locus will change accordingly with the treatment that you did. It's for theory, I don't remember the name of the molecule, I beg your pardon. However, we switched to that exactly for this specific reason. SHIP-seq is a bit more accurate and allow you to identify SNP, which will actually also three-dimensionally change the structure. For instance, for Braveheart, it's been used the same approach for identify functional element of Braveheart, which then allowed to identify HMRP-N as one of the main interactor. And that was the logic, like try to identify SNP which can actuate the structure, see if you can see an alteration in regulation, and on top of that, analyzing how can we see the same, if we can having a SHIP-seq which actually is similar to the one we did. That's why I was doing in human, because if you think about the human one, it's shorter, and then for the SHIP-seq should actually give you a more appropriate three-dimensional structure.
Okay, but you expect also to see polymorphisms or SNPs in the human sequence. So I mean, we have way more polymorphisms than mice, so how does that impact the prediction of the structure?
I think it does, okay, it will actually having way more impact depending to the sites of your null encoding, because in human we have 573 nucleotides, but if you think about a bigger null encoding, for instance, PVT1, which is actually 3K, the number of variation SNP will actually alter significantly the three-dimensional structure because you can accumulate more SNP over time. But in a shorter sequence like Bigheart, the number, or at least the likelihood that you get enough SNP in those very short sequence will be lower than for bigger null encoding. So that's why for several null encoding is not used shape-seq anymore, because realistically it's too big for being realistically sure that that will be the three-dimensional structure.
Okay, thanks. So I have another question, always technical, which regards the iPS cells-derived cardiomyocytes that you use in chapter 3 and in chapter 4. So I'm curious to know which protocol did you use. So was it based on retinoic acid, on activin A, or BMP4? So do you remember which one you used?
So also that is being done in collaboration, and one of my colleagues actually differentiated the cell himself. I did differentiate the cell once, but I don't remember fully the protocol, I have to admit. However, no, I don't remember the name of the protocol. I beg your pardon.
Never mind. But do you know at least which kind of cardiomyocytes you obtain in your after the differentiation? Were they more atrial? Were they more ventricular? Was it a mixture?
If I do remember correctly, they should be more ventricular, but I might be wrong on that.
Okay, so imagine that it's a mixture, okay, of cardiomyocytes, so atrial and ventricular, okay. How would you relate the results that you obtain in this mixed culture with the animals that you analyzed? So, for example, in the heart from the mice, did you focus just on the ventricles? Did you analyze the heart in toto, including also the atria?
No, most of the experiments were done in toto with the full tissue, so without differentiation between them. However, an interesting point is that Bigheart is expressed only in cardiomyocyte and is not present in fibroblast or other subspecies. So although the difference could be relevant in a more three-dimensional structure, we also know that Bigheart is expressed in the earlier stage of differentiation. The differentiation in ventricular atrial is came afterwards, and we already see that Bigheart starts to drop in that stage. So the most important point of our analysis was actually in the earlier stage of Bigheart, which the differentiation between atrial and ventricle is not relevant yet. If you want to study a bit more precisely that, then yes, of course, you need to actually verify that in ventricular and atrial and do the whole analysis. But the point was actually the earlier stage for us. Like that was the main point they were looking for. Okay. However, if you do want to do that a bit more properly, silence Bigheart and then differentiate in all the different aspects will be the way to go.
Okay, thank you. Do I have time for another short question? Yeah. So on chapter 3, you looked for GWAS data to assess the association between genomic variants in Bigheart and dialytic cardiomyopathy patients. But you didn't find any association, so you concluded that genomic response and not the genetic variants in Bigheart can determine the hypertrophic response. So my question is, do you think that dialytic cardiomyopathy samples are the best ones that you could have used, or because you are studying hypertrophy, and dialytic cardiomyopathy is not the genetic cardiomyopathy with the strongest hypertrophic phenotype? And first answer this question.
Sure, of course. Okay, yes, you're right. The model is not perfect. However, considering what we found later on about the mechanistic effect and the fact that, as I will tell you earlier, it's very short, so the likelihood to accumulate a lot of mutation in that area is limited compared with bigger null encoding, suggested that if we found a specific SNP which will completely alter its phenotype, it will be also like mirrored on the structural level, which we didn't see it. Therefore, we concluded that there are not specific SNP associated with this region. Therefore, it has to be associated with this three-dimensional structure. It's not 100% accurate, because in that case, you need to take from several patients, running with them, and see if you have an accumulation of SNP between all of them. However, the first evidence as a sort of like proof of concept, they say that is not the way to go. It's more associated with this structure than on its genetic locus.
Okay. But then would you expect that if a patient that harbors a mutation or a rare variant in that specific genomic region, would you expect also a change in the Bigheart?
I think it could, if you're going to alter the three-dimensional structure of it. If you're going to alter its functional elements and it doesn't take anymore the right or the appropriate shape, it might influence that. However, the likelihood that a single modification in a single area will change a three-dimensional structure is less likely. In fact, null encoding are less conserved on sequence-wise, but they are very conserved on structural-wise. And usually you have a replacement with an amino acid which has very similar function. So the likelihood that a single mutation will completely, unless it's not a codon stop, of course, but the likelihood of this is very low, because of course it's a bit more redundant. So.
Okay, thank you. I think I'm done, so I give the word back to the prorector.
Well, thank you very much, Dr. Calore. The opposition will now be completed by Professor Lumens. He's a professor of biomedical engineering at this university and also appointed secretary to your promotion committee here today. Professor Lumens.
Thank you, Mr. Prorector. Dear candidate, congratulations with this awesome thesis. I would say it looks great. I'm very impressed by the figures as well. They're very colorful and very aesthetically very beautiful. The work is very complex to me. I'm a biomedical engineer. Please consider me a plumber also when you're answering my questions. Congratulations to your supervisory team as well. I was reading your impact paragraph. Did you like to write the impact paragraph?
Yes, I did actually.
Yeah? I can read that actually, because there is millions, even billions, being predicted by you as a commercialization potentially of Bigheart, which I really like. Of course, I would like to exchange a little bit with you on and zoom out. So you state that our evidence suggests that this long non-coding RNA has a crucial role in leading to maladaptive hypertrophy. What is your definition of maladaptive hypertrophy? And please consider I'm a plumber, so I'm interested in the pump function of the heart. Why is it maladaptive?
Hi, Simo Point. Thank you for your word. And as you were saying, yes, I really enjoy write that part. Let's say that, okay, a bit of background. That paragraph is being written in the beginning of COVID, so I will say 2020. So we're like 2-3 years earlier. And the reasoning why I was written, because at the moment I was considering a study maladaptive and adaptive hypertrophy. Now, recent studies show that adaptation of the heart, it doesn't really exist because the silencing of specific gene doesn't actually improve the performance of the heart upon that. So the definition of adaptive and maladaptive might be a bit outdated. However, the logic of maladaptive, if we can consider in the broader term, will be when the heart starts to grow. Let's say that when you have a stress, the heart will try to grow. That's logical because, of course, you need more strength for pumping the heart throughout your body. For instance, pregnant woman, they actually almost double the size of the heart because, of course, it's necessary. Athletes show that they having a slightly higher, bigger heart, but it's still performing well. And that's what I was considering adaptive hypertrophy. However, the difference, I think, is actually in the three-dimensional structure. Because what the heart does is pretty much increase the amount of the chamber, so it can actually, more blood can flow in, and increase the strength of the muscle around, so it can push it around a bit more properly. However, when this is actually overdue, even if you have too much dilatation of the chamber, too much liquid get inside, but I don't have enough strength for put it outside with the same intensity, or, and this is an eccentric hypertrophy, if I remember correctly, instead the other version is that you have a very thicker layer of muscle, but it's so thick that the chamber actually is less big, therefore, even if it's actually pushed stronger, it's still less performing because I push less blood over time, so I need to beat more. And as I was describing as a power lifter, as much you can power lifting as much as possible, at some point you will start to get weaker. So in that case, I will consider that as a maladaptive adaptative. But of course, it depends a lot from the case, because you need to also consider what is called ejection fracture. And we know there are 3 different kinds now: conserved, well, preserved, restricted, and an intermediate phase. And that, I think, is going to be the new definition when you think about cardiac hypertrophy, how your heart was still able to carry its function properly or not.
Okay, so from your answer, I can already deduct there is a whole spectrum that can happen. And maybe from the big hearts, it may be a problem with the filling of the heart, or not?
we did investigate this particular aspect. We verified in our heart which we induce cardiac hypertrophy. And if I remember correctly, those models induce pressure overload, which is actually the one with the higher amount of flux, but less strength to go around, because of course pressure overload will be that. However, we could also try to verify if you have a different set. I don't expect huge differences, because I think Bigheart is reactivated as a part of like a progress program, which is not necessarily anymore in fully differentiated heart, and reactivate upon that. However, we could actually see a different spectrum of isoform of Bigheart, which could still make a role there.
Okay, so if I may take you to the follow-up sentence, we also provide the evidence that the modulation of long non-coding RNA can
Dear candidate Nicolò Mangraviti, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. And I request you and your company to await the results of our deliberations here in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
To the west side, no place like home. If it's questions that you've got, go the extra mile and die. I know what's on the line. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like, my goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Traveling so far from home, gathering knowledge, other connections. I put the time in to get everything I have home for. It's a challenge and we all in. It's a better future than we go for. Even though these nights seem long, I know that I am never on my own. Know you won the battle once you grow. Southside, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and die. Can't fly by now, don't waste waste all your time, 'cause I'll go the extra mile.
Dear candidate Nicolò Mangraviti, the degree committee here present has assessed the quality of your thesis and its defense. And in view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor De Wind is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Please all rise.
Dear candidate, I would like to ask you a question first, and let's see how you answer to that, yeah? Do you promise to work in accordance with the principles of scientific integrity, at all times to be careful and honest, transparent, independent, and responsible? What is your answer to that?
I do promise.
Very happy to hear that, because then by the authority vested in us by law and conformity with the decision of the committee here present, I hereby confirm upon you, Nicolò Mangraviti, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now, in 2 seconds, will present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. And we do that now.
Cheers.
Please be seated.
Yes. Well done. It was quite a journey. But I'm very happy that I'm the first one to be able to call you dottore, doctor, and to congratulate you. And I would like to just use a few words to finalize and celebrate this special day, where we also include your new wife and your family, your father, mother, I think it's your brother, and the in-laws. So congratulations to all of you. So let's go back a little bit. Yes, Nicolò entered our lab as a young, quite talkative man from Sicily, and you soon very easily integrated in the group, which was helped by your social abilities. I think we all witnessed today how easy you can use your mouth, so there's no problem there. And you have an extremely overall joyous nature. Yeah? You always see the fun in things, you always laugh, you always make jokes. As well as the fact that and of course, the fact that you could integrate easily is also you entered into a group with many internationals. Vast majority was Italian, so it was almost as if you got home away from home. So I think you really felt at home quite quickly. And soon enough, you felt so much at home that you actually became some sort of a driving force, or maybe we could use the word force of nature, behind a plethora of social gatherings and evenings out, and even travels abroad with your PhD colleagues. And you showed off your abilities. This is what I heard. You showed off your abilities in the kitchen, so you're actually a very good cook as well. And regularly displayed your medals of bravery in social gatherings. Yeah. So one example that I experienced was quite remarkable. So on a regular Monday, just as any other Monday in the summer, I came to work. And then I see Nicolò entering the lab with huge scars in his face, bruises and stitches, and it was horrible. It's like he was ran over by a truck. After an episode where you jumped into a pool, a public pool, I heard, and then you hit your face on the diving board, yes? Yeah, side of the diving board in a public outside swimming pool, leading to a massive amount of blood in the pool, so nobody could enter the pool anymore. It was as if there was a piranha attack. So people screaming around, and everyone in disarray. A trip to the emergency room, and yeah, of course, these beautiful bruises and scars and stitches for weeks in your face to show off that medal of bravery. So I think we could all notice that Nicolò's presence from far in streets. I once in a while met you also in the city center, and I could immediately recognize you from your presence, wearing always a hat. So a very fashionable hat, and yeah, inside buildings you could be recognized by just the loud voice and the laughters, and this is where the fun was happening. So you could easily recognize where Nicolò is. So nothing human is strange to PhD students, nor Nicolò. Where in 4 years a PhD student or a contract, you basically, what I notice now over all these years is that the first 2 years, PhD students just take their time, they're getting very comfortable, so basically they have no sense of time passing, although the time passes just similarly, yeah? But the sense of time is just, we have all the time in the world. So it's characterized often by a sense of aimlessness and procrastination somehow. So this is general to all PhD students, and then only that is followed by by the slow seeping in of the realization that writing a thesis and preparing yourself for the next career step is actually the whole goal of getting a PhD, of this certificate. So that seeps in very slowly, and it somehow, suddenly there's no more time for social gathering. There's a bit of an urgency, there's always panic sometimes. And the closer you near to that end date, this particular date that we are now, this panic then accumulates and accumulates, although time passes exactly with the same pace. So it's really funny for us supervisors to see that. But so the time perception for PhD students is completely different, accompanied with a lot of emotion. So, so pressure inside is then translated towards the outside, which is logical. And then the favorite objects of that pressure and to blame are the supervisors, yes? So Nicolò certainly also had that sense of extreme level of panic and anxiety, but yeah, from a supervisor's bird-eye point of view, time keeps passing with the exact same pace, and as always, we know that a thesis will be produced, and in your case, also a thesis was produced. Well, you certainly increase the pressure for yourself by, towards the end, by deciding to do everything at the same time. Really big life decisions, getting married, traveling to the United States, which is actually a very smart idea of you, and I really compliment you on that. While also at the same time finishing a thesis, while we're still roughly in a sort of a COVID or a post-COVID period. So it was like an explosion of everything that could go wrong in the sense of writing a thesis. But I'm here to tell you that you did it. This certificate is the proof of it. I really am proud of what you did. I see similarities between us. Me also, just right after my thesis period, I went to the United States to do a postdoctoral. It was the most beautiful period in my life. So I really would love for you today to enjoy joking around with your former colleagues, just being happy, enjoy the one last day today, and I wish you the very best in the future, wherever that might be. in whatever continent that might be. And I hope you will always keep doing that in your own unique way. Yes, so wearing a fashionable hat and making sure that everyone in a room knows you're there with your loud jokes and thunderous laughters. Yeah, so congratulations again and enjoy today.
Thank you, Professor De Wind. Dear Dr. Mangraviti, and also on behalf of the Maastricht University, I congratulate you with the degree you have acquired. And with this congratulations, I would also like to include your promotion team, of course also your family and friends and colleagues. The committee was impressed with your defense. You were knowledgeable and confident, and you were also born with a talent, with an exceptional talent to explain complicated scientific matters to society. And for that, I think we should also thank your family and your brother, Marco, for raising you in such a way. For now, I would like to thank the external opponents for being here online today. And I would like to ask all your colleagues who already leave for the rafter after we finish this ceremony, and ask the family to stay behind and the photographer for making some pictures here. And then we'll see the rest of the audience in the rafter. And with that, I'd like to close this academic ceremony.
Thank you.
Very well done.
Thanks so much.
Yeah, really good.
Move a little bit.
Congratulations.
Thank you.
Very well done.
Thanks so much.
Red Sea here.
It's wonderful.
Please come forward, Nico. Yeah. Para nims.